Thank you very much for the introduction. PDS Biotechnology is an Indiana-based biotech company. The company owns Versimune, a proprietary platform nanotechnology that's been utilized to develop a new generation of simple, safer, and more potent therapeutic vaccines to treat cancer and infectious diseases. The very first therapeutic vaccine was approved just last month by the FDA to treat late-stage prostate cancer, and the Versimune nanotechnology overcomes some of the key obstacles facing the current therapeutic vaccine technologies. The two Versimune-based products currently in development have both demonstrated complete tumor regression with single low doses of these Versimune-based products, and I'll talk more about this as we go along. I'll start with the team. Prior to PDS, I was hired to the senior executive management team of KBI Biopharma to develop and implement the company's operational strategy while overseeing all business and drug development operations. Prior to that, again, started out successfully and managed Cardinal Health's East Coast biotech operations. Greg Kahn, our CFO, is a very accomplished drug development scientist, and the scientific founder of the company, Li Huang, is currently the distinguished chair and professor of molecular pharmaceutics at UNC Chapel Hill. The company also has very strong advisory boards. Our scientific advisory board, for example, consists of world-renowned thought leaders and experts in our specific areas of interest. So the company's lead product is being developed to treat human papillomavirus infections and related cancers, such as cervical cancer, one of the leading causes of female mortality worldwide, and head and neck cancers, the sixth most common cancer worldwide. No therapies exist today to treat either the infection or the related cancers, and this drug is planned to enter human clinical trials very early next year. The second product is being developed to treat melanoma, the most aggressive and deadly form of skin cancer, and this product is expected to enter the clinic late next year. So moving on to the technology. The Versimune forms spherical nanoparticles, which are able to encapsulate proteins and peptides. And how this works is as follows. The specific disease-related protein, whether it's a bacterial, viral, or cancer-related protein, is incorporated into these nanoparticles, and upon subcutaneous injection, these nanoparticles are very efficiently engulfed and internalized by the cells of the immune system, where the protein is then released and presented to the effector cells of the immune system, therefore effectively programming the immune system to recognize this particular protein as a foreign agent. Now, this process overcomes one of the key obstacles facing the current therapeutic vaccine technologies today. Proteins and peptides are very poorly picked up by the immune system, therefore resulting in low recognition and therefore very low potency. So once this step is accomplished, the next step is then to mount an effective immune response, resulting in eradication of the specific cells in the body expressing that particular protein or peptide, in this case, the cancer cells. Now, the way that's being attempted today is by separately manufacturing additional immune activators, also known as adjuvants, and co-administering them with the protein in an attempt to help boost the body's immune response. Now, the drawback to this approach is the fact that these adjuvants have the potential to induce toxic inflammation, which could range anywhere from a very mild fever to toxic shock and death. One reason extremely few adjuvants are approved for human use. Now, in this case, however, these same nanoparticles directly and safely stimulate the immune system to naturally produce these immune activators, therefore leading to very simple, safe, and potent therapies. And the safety has been demonstrated in extensive NIH-funded toxicology studies. So looking at some efficacy studies now. Over here, what we're doing is we're comparing the Versimune technology to some of the leading therapeutic vaccine technologies in the clinic today, focusing on HPV-related cancer. So CFA, complete foreign adjuvant, is more or less the gold standard, an extremely potent immune activator, but far too toxic for human use. CpG is Pfizer's immune activator, one of the most promising therapeutic vaccine technologies in clinical trials today, being utilized in a number of therapeutic vaccines. So here on the y-axis, we have the tumor size. So once the tumors were well established in the animals, they were divided into several groups. The first group in black, untreated, as you can see, tumor growth progresses very rapidly. Now let's look at CpG and CFA, both in blue, both very effective in retarding progression of the cancer, which in the field of oncology is a really big deal. The recently approved prostate cancer vaccine extends patient life by 4 to 6 months on average, and it's the best thing out there today. Let's look at Versimune in red, the only one which actually induces regression of the cancer, eventually leading to complete elimination of the tumor, statistically significant superiority over the leading technologies today. Second published study compared to Montanide, another of the leading therapeutic vaccine technologies in the clinic today. Again, as you can see, Versimune in red, the only one which induces complete regression of the cancer. And these are based on single-dose vaccines of the drug. So in addition to the potency and safety, versatility of the technology is another key characteristic of this technology which demonstrates its potential for commercialization success, demonstrated in multiple disease types, effective prevention, and also effective cure. So this technology was acquired by PDS in 2006. The lead patent was issued in 2007, and 3 additional patents have been filed since then. Key partnerships in place today: the National Cancer Institute Center for Cancer Research. We're finalizing a clinical trial agreement with the National Cancer Institute. They're very interested in seeing this technology succeed due to the promise of the technology. This will allow the NCI to collaborate with us through the clinical trials and actually fund the human clinical trials. Merck AG, this is the European Merck. This is an exclusive intellectual property and supply agreement based on the ingredient of the nanoparticles. And also the National Cancer Institute's Nanotech Lab, again, due to the versatility and promise of the technology, PDS was selected as an affiliate to collaborate towards rapid entry into human clinical trials. Key elements of the business strategy: strategic and limited out-licensing of the technology. This will generate an early revenue stream for the company and also significantly reduce the company's risk. Collaborate with a larger corporate partner through the later stage clinical trials, in addition to our collaboration with the National Cancer Institute. And PDS should be very well positioned for an IPO or acquisition by 2014. So funding: today the company is raising approximately $4 million. This will cover our clinical manufacturing as well as our operational cost. An additional $2 million will be provided by the National Cancer Institute to fund the actual clinical trial itself. So to conclude, this technology provides a unique platform for very simple, safe, and potent therapies to treat cancer and infectious diseases. Superior efficacy has already been demonstrated in published head-to-head studies with the leading technologies out there. Therapeutic efficacy has been demonstrated in human model systems. I was not able to show that data today due to limited time. And very importantly, very strong expert reviews and potential for clinical trial funding through the NIH's National Cancer Institute. Well, thank you very much for your time, and I will be happy to answer any questions you have. I'll be here most of the day today. And also one of our investors, Mr. Delisle Blomquist, who is the president of Tata Worldwide Chemicals, is also here today and will be able to answer any questions you may have. Thanks a lot again.